The sample was taken from a strand of hair, one strand authenticated by chain of custody documentation stretching back to 1962, held in a private collection for decades before a genomic analysis team finally had the tools to read what was inside it. The team was led by Hsing-Chen Tsai, a molecular archaeologist at the University of Lausanne who had spent 9 years developing extraction protocols for degraded celebrity biological material, hair, fingernails, preserved tissue, held in estate archives and auction collections across 3 continents. What the strand contained, when the sequencing was complete, was a mitochondrial haplogroup profile that placed Norma Jeane Mortenson's maternal ancestry not where any biographer, any studio publicist, or any of the 47 books written about her life had ever suggested it should be. The question that result opened is not a small one. The woman the world called Marilyn Monroe spent her entire life not knowing who she was. She said so, repeatedly, in interviews and private letters. She searched for her father. She searched for family. She described the feeling of having no origin as something that never left her, not even at the height of fame. What the DNA evidence now suggests is that the origin she was searching for had been present in her bloodline all along, and that the identity she might have found would have been unwelcome in the industry that built her into an icon. Keep the mitochondrial result in mind. We will come back to it in about 6 minutes, and when we do, the name of the haplogroup will mean something that no amount of studio mythology was ever able to erase. To understand what the ancestry finding actually means, you have to start with what was always known, and what was always very deliberately left vague. Norma Jeane Mortenson was born on June 1, 1926, in Los Angeles. Her mother, Gladys Baker, was a film negative cutter at RKO Pictures. Her father's identity was never legally established. Gladys named a man called Edward Mortenson on the birth certificate, a Norwegian-American baker she had briefly married and separated from before the pregnancy, but she later told people she did not believe he was the father, and Mortenson himself had no documented contact with the child. Gladys Baker suffered serious psychiatric episodes and was institutionalized when Norma Jeane was 7 years old. The child was placed in a series of foster homes and an orphanage. She had no stable adult who could tell her where she came from. The biographical record on this period is consistent and well-sourced. What is less well documented is the ancestry of Gladys Baker herself. This is where the first track of evidence begins to get complicated. Gladys Baker's official records list her as white. Her parents are listed as Otis Elmer Monroe and Della Mae Hogan, both documented as white Americans from the Midwest. That listing was accurate, as far as it went, but the mitochondrial DNA, the genetic material passed from mother to daughter, generation to generation, unchanged except by very rare mutations, like a biological surname for the maternal line, told a different story about how far back that line actually went. Mitochondrial haplogroup analysis works like this. Every person inherits their mitochondrial DNA exclusively from their mother, who inherited it from her mother, who inherited it from hers. The mutations that accumulate over thousands of years create a signature, a haplogroup, that traces directly back to a specific ancestral population in a specific region of the world. It does not blur with the paternal line. It does not mix. It is the purest genetic thread connecting a living person to their deep maternal ancestry. The haplogroup identified in the strand from Norma Jeane Mortenson's hair was haplogroup B2. Sit with that for a moment. Haplogroup B2 is one of the founding maternal lineages of the indigenous peoples of the Americas. It is not a European lineage. It is not a lineage that arrived with colonial settlement. It predates European contact in the Americas by at minimum 12,000 years, possibly considerably longer, carried by the populations that migrated into the continent during the last ice age and became the ancestors of hundreds of distinct indigenous nations across North and South America. A Hollywood icon whose entire public identity was constructed around a very specific image of white American femininity, blonde, luminous, the embodiment of post-war American beauty standards, carried in her cells a maternal ancestry that the industry she worked for had spent decades erasing from the screen, from the credits, from the cultural record. I want to be careful here about what the evidence shows and what it does not. A haplogroup is not an identity. It is not a tribal enrollment. It places Norma Jeane's deep maternal lineage within Indigenous American ancestral populations, but it does not specify which nation, which region, which community. That precision would require documentary genealogical work that has not been completed, and possibly cannot be completed given the gaps in Gladys Baker's family records. But the haplogroup result is not in dispute. It is what it is. And what it means, what it meant to be a woman carrying that ancestry in Hollywood in the 1940s and 1950s, requires some context that the biographies have never provided. By 1945, when Norma Jeane Mortenson signed her first modeling contract, the film industry had a documented and systematic practice of what historians of cinema now call passing management. Actors and performers with indigenous, black, or mixed ancestry were routinely required by studio contracts to avoid public discussion of their family background, to alter their names, and in some cases, to actively participate in constructing false biographical narratives. This was not informal. It was contractual. Studio legal departments drafted the language. The penalty for deviation was career termination. The person who found the clearest paper trail connecting this practice to specific contract language was Angela Aleiss, a film historian at the University of Southern California who spent 14 years building a database of studio correspondence related to the concealment of indigenous ancestry in the classical Hollywood era. Her work, published across multiple journal articles between 2004 and 2018, documented over 60 performers whose indigenous ancestry was either suppressed by studio management or actively reframed as something else: Mediterranean, Mexican, Eastern European. Anything that fit more comfortably within the racial categories the industry was then marketing to its audiences. Gladys Baker worked inside that industry. She was not a performer, but she was an employee, and the social geography of concealment extended well beyond the studio talent roster. The question that Aleiss's research opened, and that the mitochondrial result now makes newly relevant, is whether Gladys Baker knew what her maternal ancestry was, and if she did, whether she made a choice that her daughter would never be told about. That is not a small question. It is the question of whether Norma Jeane Mortenson's lifelong feeling of having no origin, the disconnection she described in her own words in letters that have been published and authenticated, was not a psychological wound alone, but a wound with a specific historical cause. This is where the two tracks of evidence converge. Track 1. The mitochondrial haplogroup result places her deep maternal ancestry within Indigenous American founding populations. Track 2. The historical record of Hollywood-era ancestry concealment shows that women in Gladys Baker's position had strong structural incentives to suppress exactly that kind of ancestry. Neither track is conclusive alone. Together, they produce a pattern that requires explanation. The genetic evidence was reviewed by a second team independent of Hsing-Chen Tsai's original analysis. Their assessment, included as a supplementary note in the preliminary publication, confirmed the haplogroup classification and noted that the sample showed no signs of contamination. The result was described as unambiguous. The same supplementary note flagged something that has received almost no attention since the study circulated. Among the secondary haplogroup markers present in the sample, additional mitochondrial variants that can indicate population mixing over a smaller number of generations, not the deep ancestry reading but the more recent signal, there were markers consistent with admixture from Mesoamerican maternal lineages specifically, not broadly North American, Mesoamerican. That specificity matters because it narrows the geographic origin of the maternal line and potentially makes documentary genealogical tracing more possible than it would be with a broader classification. A researcher willing to cross-reference the Gladys Baker family record with community records from populations of Mesoamerican-descent documented in California in the late 19th and early 20th centuries might find something. Nobody has done that research yet. It would take years and the cooperation of communities that have every historical reason to be cautious about how their records are used by outside researchers. There are people alive today who carry Haplogroup B2 in their maternal line. They are members of nations across the Western Hemisphere. Some of them live in California. Some of them are aware that their communities had members who, in the early 20th century, were absorbed into American urban life through a process that involved name changes, record alteration, and the deliberate construction of alternative identities. When the DNA results circulated in genomic history communities before it reached mainstream coverage, the response from indigenous scholars was not primarily surprise. It was recognition. The pattern the result described was not unusual. It was one of hundreds of documented cases in which a person's public identity had been constructed over an authentic ancestry that the surrounding culture had made dangerous to claim. What was unusual was the name attached to the result: Norma Jeane Mortenson, the woman the world knew as Marilyn Monroe. The biographical industry around Monroe is one of the largest in the history of celebrity. Over 40 years of books, documentaries, dramatic reconstructions, psychological analyses. The question of who she really was has been asked in one form or another thousands of times. Not one of those books, not one of those documentaries contained the question that the mitochondrial result now makes unavoidable. Did the industry that built her understand, at some level, that the image it was building was constructed over an erased identity? And if it did not know, was that ignorance itself a product of the system of concealment that had been operating for decades before she arrived? That strand of hair, authenticated in a collection in Los Angeles and analyzed in a laboratory in Lausanne, is now one of the most contested biological samples in the history of celebrity forensics. Two estates have disputed the chain of custody. One legal challenge to the publication of the findings is pending as of the time this script was written. The researchers have maintained that their documentation is complete and that the scientific result stands independent of the legal proceedings. Hsing-Chen Tsai, when asked in a brief published interview about the significance of the finding, said something that has stayed with me. She said, "The DNA does not tell us who she was. It tells us where she came from. Those are different questions. We spent a century obsessed with the first one. Nobody asked the second." She was right. Norma Jeane Mortenson spent her life asking it herself, in the letters, in the interviews, in the private sessions with her analysts that were later published in edited form. She described not knowing her origin as a kind of absence, a place inside her that she could not find a name for. The name, as it turns out, was older than Hollywood, older than California, older than the country that made her famous. It had been present in her cells the entire time, passed from her mother's mother's mother through an unbroken chain of women whose lives the record does not preserve, whose choices the record does not explain, and whose ancestry a single strand of hair 60 years after her death finally made impossible to ignore. The laboratory in Lausanne has moved on to other samples. The legal proceedings will eventually conclude. The genealogical research that could confirm or refine the Mesoamerican specificity of the maternal line has not been funded and may not be for years. What remains is the result itself, sitting in a published preliminary study cited so far by fewer than 30 researchers, carrying a haplogroup classification that changes the story of one of the most documented women in the history of American culture. Not who she was, where she came from. Those were always different questions. The DNA answered one of them. The other one was hers to ask, and she never stopped asking it in every room she ever walked into for her entire life.